My name is Manoj Lalu again, and I'll be presenting just a bit of an overview of what we're doing with this GoCART program. GoCART is just a cute acronym for this Getting Better Outcomes with Chimeric Antigen Receptor T-Cell Therapy. So this is a program that BioCanRx has actually funded. In terms of my background, and I should just mention that if you don't know what CAR T cells are, don't worry. I've got a slide a little bit later on explaining it a bit more as well. So just a quick slide about my own background. I'm an assistant professor and associate scientist in the Department of Anesthesia and Pain Medicine. I'm also part of the Clinical Epidemiology program and the Regenerative Medicine program at the Ottawa Hospital Research Institute. So clinically, I'm actually an anesthesiologist. I deal and treat patients with cancer not infrequently in the operating room, and also patients who have unfortunately have cancer and have complex pain issues. We're the folks that deal and help treat those issues as well too. And just as a caveat, most of my training has been at the bench in terms of lab work actually, and just over the last couple of years I've become more involved in the whole trial side and the whole clinical epidemiology side, and that's where I really became interested in this because we do all this fantastic work at the lab and we don't always see it instituted at the bedside. So I was very interested in the whole process of what we call bench to bedside translation. And we know that this sort of translation is slow and ineffective, as people have already mentioned in previous presentations. And in fact, I've sort of described it here as clinical translation. This whole process from bench to bedside is arduous and it's highly error prone. So when people have looked back at what we call high impact discoveries, so at the bench, so these are the things that you hear about, on CBC News. X, Y, and Z lab in Vancouver found this fantastic finding in mice, and they think it's going to affect health downstream. When people have actually looked back at this and they said, okay, well, how many of those high impact discoveries, those sort of discoveries that created a lot of buzz, got a lot of media coverage and were in sort of more widely read journals, how many of those actually were translated? And these are discoveries that also claim to have some clinical relevance as well. Well, it's only 5%. So 1 in 20. So that means 19 out of 20 of those fantastic discoveries don't actually end up affecting patient care at all. And this is talking about within a decade. What's interesting is for those of you that sort of follow the whole oncolytic virus literature, TVEC, which is an oncolytic virus therapy, the first and only one that's been approved so far by the FDA, took 15 years from the point where that first paper was published to FDA approving it last year. So it's a very, very long process. And then on top of that, we start thinking about, okay, those agents that actually do make it to that first clinical trial, how many of those actually are ultimately licensed? Well, only 11% of those are licensed in the end. So that means 9 out of 10 of those first-in-human trials, or those agents, they're failing. And this is a huge problem because such large-scale attrition, it's potentially harmful to individuals in trials because, number one, they are either being exposed to treatment that doesn't work at all, or even worse, they might be exposed to treatment that causes harm. And on top of that, it's consuming these really scarce human resources in terms of our research assistants and our research nurses and our clinicians that are involved in these trials, and also our material resources. As I'm sure we're all aware of in this room, research funding is quite scarce, right? So we have to decide where we're going to put our dollars. And ultimately, the cost of all this failed translation results in higher drug costs because drug companies and other entities have to recoup the costs of these 9 out of 10 trials failing, right? So how are they going to do that? Well, the few that actually make it through this pipeline ultimately have higher costs as well too.
Can you talk a little more about why you think we have this only 11%? Is it because the trials aren't properly designed? Is it like...
We'll get to that. That's what the whole GoCART program is about. So, it's multifactorial, as you've already started to point out there, and I'll get to that in a couple of slides, actually. Okay. So in terms of thinking about this, how this looks, sort of picture, on one side we have folks like myself and others who work at the bench here, and they've got these fantastic discoveries. And on the other side, we have our clinicians and patients there anxiously awaiting these new therapies. However, it's a very, very rickety bridge that goes across that. So what GoCart is trying to do is try to strengthen that bridge and try to address why is that bridge so rickety, and how can we actually strengthen that? So before I get into GoCart and how we're starting to think about, sort of address some of these issues that are arising, just one brief slide on CAR T-cell therapy. So CAR T again stands for Chimeric Antigen Receptor T-cell therapy. And just really briefly, it's potentially a new and very powerful tool to treat blood cancers. So just in this cartoon here, let me see if I can find the cursor here. Okay, yeah, so just in this cartoon here, we have our patient who unfortunately has this blood cancer that's been refractory to the normal sort of therapies. So what they've been able to do is take white cells from these patients, so that's this leukapheresis here. So taking white cells from these patients, these white blood cells that fight cancers, that they fight infections as well. They take them from these patients, they put them in, get them in the lab, and then they do what's called transfection. So they transfect it so that these cells, these white cells, this red cell here, is now able to express this receptor. This receptor will help the body identify and fight the cancer. Basically from there, they grow these cells up and they activate them. So they're activated T cells. They do what's called expansion, so they grow them up to larger numbers. And then ultimately what they do is they infuse the patient's cells, which have now been modified, back into their body. And what's been very remarkable is that in the small number of trials with small numbers of patients that have been completed so far, they've had some basically cures. So patients who would otherwise would have died within a few months to a year now are in remission. But on top of this, what's complicated all of this are a number of trials where they've had some very serious adverse events. There's been one trial that was stopped by the FDA in the last few months where they actually had several treatment-related deaths. So deaths that were immediately related to the CAR T cells themselves. So as you can already start to imagine, there are a number of complex potential issues that are around this therapy. And again, what our BioCanRx is invested in and what our group is invested in is to try to bring CAR T-cell therapy in a trial format, investigator-led, to Canada. But before we do that, we decided to try to do our homework, as Justin has already alluded to try to start to identify these complex issues and also to try to start to address them as well. So as I've already mentioned, there's a number of adverse events in trials to date. So what is our understanding of that? We're going to try to understand those better, try to understand how we may be able to avoid them, how we may be able to identify patients who might be susceptible to those sort of adverse events. Next, as you can imagine, these sort of cell therapies are very expensive. So how are we going to make this affordable to our publicly funded healthcare system? How can we design our trial and the patient eligibility for this to make this more cost-effective and feasible for this healthcare system? Next, as Justin has alluded to, how many patients would be eligible? This is a huge problem in first-in-human and early-phase trials. Investigators say, I've got this fantastic therapy, it's going to work, people are going to flock to this, and we're going to get so many patients, we won't know what to do, we're going to be able to finish our trial in 1 year, I'm going to get funding for 1 year, get it done. 5 years later, they've recruited 5 patients, and that's an actual cell therapy trial for MI, for myocardial infarction. So, and they're looking for 10 patients. Right, so this is a very real problem, and when you start to look at the numbers around this, 80% of trials, and this is not just cell therapy trials, this is actually all trials, fail to accrue, that means recruit, the number of patients they expect when they write that first initial grant and say, okay, in 5 years I'm going to get X amount of patients. 80% of them fail to do that. And this is a huge problem, and no one's really looked at this very carefully. And we start thinking about what we're talking about, which is very expensive, to start to generate all these viruses, to grow these cells up. to have a trial run for several years longer than you expect, it can be cost prohibitive and may even cause a trial to fold because you won't be able to basically finance a trial for that long. So in order to address all this, as you can imagine, and these are just a few of the issues, you need expertise from clinical epidemiology, you need expertise from health economists, you need expertise from basic scientists, you need expertise from implementation scientists, like Justin. You need expertise from the hematologists who are treating these patients day in and day out. And then very importantly, I've underlined it here, you need input from patients to help identify some of the solutions to these concerns that are arising in multiple first-in-human or early-phase trials. So just to break down how we've started to think about this into 4 points and solutions on how we can bring CAR-T therapy safely and effectively, again, to Canada as an investigator-led trial. And this is what GoCART is really all about. First of all, we've said that we need a better understanding of the preclinical work. And I know most of you are patient partners or patient advocates, so it may be surprising to you that most first-in-human trials are conducted without systematically summarizing and assessing all the preclinical work that's been done. I can say this safely because we were the first group to do that, not for CAR T cells, but for stem cell therapy for septic shock. This is highly problematic because we know, looking back at some failed trials, that if a systematic summary had been performed of preclinical work, they probably shouldn't have done the trial because it was basically justified by saying, there's a fantastic paper in Nature and Science. These are very high impact journals. It shows this therapy works on 30 mice per group. We should proceed to trial. But then when we start to look at work that was published in lower impact journals, maybe the ones that didn't receive all the buzz that 1 or 2 papers in Science and Nature received, you could see that the effects of whatever drug or intervention you're looking at weren't as promising. And in fact, when you quantitatively look at this, the effect is no longer there. So for CAR-T, it's a little bit of a different situation because there have been now some early phase clinical trials that we can go on, but that still doesn't preclude us from looking at that preclinical work, that bench work, to see if we can tease anything out of there that would help inform the clinical trial protocol itself to make CAR-T cells in our hands more safe and effective than it's been in previous trials. Number 2, this is this knowledge synthesis. So again, summarizing all of that work, not only from the bench, but also that early phase clinical trials that have been performed. So we're basically going to go in and try to find out and summarize everything very quantitatively in terms of what's been published in early phase trials for CAR-T therapy.
Can I ask a
Sure.
stupid question? Better understanding of this preclinical work, which disciplines are appropriate to do that kind of analysis?
So it's interesting. So our team, everyone's involved. So everyone, I'll get to that in a second, but it's basically health economists. So it's those folks I was mentioning before. So hematologists are involved. Certainly folks like Rob Holt, who's leading the cell production from UBC, he's involved in this as well. He's saying, okay, what sort of elements should we be pulling out? What sort of information should we be pulling out from all these things that have been published? So basically everyone has a little bit of role. Exactly, it's multi-stakeholder. That's a much better way to describe it than I just did. And you might be wondering, well, what does the health economists have to do with this? Well, we can predict some costs even from just preclinical work as well too, and we've done that in the stem cell therapy realm for septic shock, for instance. And septic shock, for those of you that aren't familiar, is just a life-threatening infection. And then finally, integration of stakeholders. So that includes patients, that includes physicians into the trial protocol development. So GoCART is not about doing the trial. It's about doing all the homework leading up to the trial. So we're taking 2 years to do this. Up to 2 years to do this, I should say. And I'm going to go into a little more detail as to what all these different components are that we're going to be doing. So again, in A here, this is just the preclinical and clinical systematic reviews. This is again just summarizing transparently everything that's been done and published so far in the CAR-T field. Second is knowledge translation. I've got a couple of slides that I'll be finishing off with talking about that, but that's basically taking what Justin had talked about in his previous presentation and applying that to the early phase clinical trial field, or the first-in-human clinical trial field. So trying to understand the barriers and enablers for number one, patients participating in a trial like this, and number two, for physicians who are on the ground who are going to basically asked to help recruit these patients. What are the barriers, enablers of that? And also these patients can get very sick, as I've already pointed out. So what would be the barriers, enablers for them providing ongoing care for these patients who receive CAR T-cell therapy? Panel C here is an early economic analysis. So basically with the limited information we have right now, we should still be able to provide an estimate of how much this may cost our health system per patient downstream. Next, we have a number of different databases that exist across the country where we have information on patients who have had blood cancers and been treated. So using that, we'll be able to estimate how many patients might roll through the different centers that would be involved in this trial, and also start to look at what are some of the adverse events in these patients, even without CAR T-cell therapy. So we have a baseline rate of how sick these patients are, even without something as experimental as CAR T-cells. And then finally, a very big portion of this is production and testing of the cell product. And again, I already mentioned Rob Holt from UBC will be leading that. It's its own project in itself. So the whole wheel here, our go-kart wheel, leads into trial protocol development, where we have, again, all our stakeholders that I've mentioned come together to help synthesize all this information to try to deliver a trial protocol. We hope to do that, again, within a year and a half to 2 years. And what's very important is this yellow line here, which represents our, well, it was an arrow, but the arrow's cut off here at the bottom, but it's integrated knowledge translation, which is really engaging our stakeholders throughout the entire process. So again, we've already talked about this a little bit, but just to reiterate what IKT, or Integrated Knowledge Translation, actually is, it's taking our stakeholders, our potential knowledge users, and having them get engaged in the entire research process. So this is true collaboration, and again, our stakeholders that we've identified are patients and physicians, and we're asking them to help us shape the entire research process. So again, this is over the next year and a half to 2 years, and this is doing all our homework again to develop this trial protocol to make it as safe and as effective, and also to address patient concerns as well, obviously. So in terms of our RKT, what we're practically doing, we've had active involvement of, again, patients, clinicians, and we're trying to get a regulator on board as well too. And we've had a patient and physician representative already involved. We're just starting this process, but already involved in the few months that we've already started this process. So we've been very fortunate to have Matthew Seftel, gentleman on the left there. He's a hematologist in Manitoba. So again, he's a clinician who deals with blood cancers and treats patients with blood cancers day in, day out. However, he's not as excited about CAR T-cell therapy as we are. So he's actually providing sort of an outside perspective from the inside, in other words, right? So sober second thought. We have obviously other hematologists who are very excited about CAR T-cell therapy, like Dr. Natasha Kekre, who's going to be leading the trial downstream. But we wanted someone to provide, again, a little bit of sober second thought to what we're thinking. And then on the right is our patient partner, and that's, for those of you in Ottawa, Stuntman Stu. He's a radio personality, actually. So Stuart Schwartz, and he's actually had a very public diagnosis of his blood cancer and treatment, and thankfully subsequently remission of his blood cancer as well too. So he's been fantastic. He's been involved in all our meetings up to date, and he's been very vocal on when we've asked him for input on various aspects of what we're doing. And I'll point out one of our sub-projects, which he's going to be very involved in, which is some of the interviews and some of the surveys that Justin had already mentioned, to try to understand what might be the barriers and enablers to running this trial. And I'll just point out really quickly, this is mainly, well, I guess it's for patients here, patient partners and patient advocates as well too, but this site within Patients Canada, there is one page where they have a couple of PDFs, which I thought were excellent, especially for someone like me who has a short attention span. Basically in point form, very quickly, what should a patient think about when they're starting to think about getting involved in this sort of research, and what should a researcher in a second PDF start thinking about when they want to start involving patients in their research as well too. So this sort of resource, along with the expertise that Justin and Zara have brought to the table, has really sort of helped shape the GoCart program. So just in the last couple of minutes, in terms of what we've done for GoCart and how we've involved Stu. First of all, how we recruited him. Well, initially it was through a physician co-investigator. And on top of that, we've partnered with the Leukemia and Lymphoma Society of Canada. They wrote a letter of support for the GoCart program. And they're going to help us identify others. And an ad has been sent out through LLS recently, so that society. And this kind of goes towards that whole thinking around, do you want a panel or do you want an individual patient? Right now it's an individual patient, just because we've started up and Stu has been on board, but we're going to try to spread out the responsibilities of what we're hoping our patient partners will do through a panel. And we've also opened it up to caregivers as well too. And again, a very important consideration, whether you just want to restrict it to patients or caregivers as well. And then finally, how to keep, how we've kept our patient partner involved. Well, number one, we've identified a point person who's been me. So I've been basically talking to him before every meeting. So we've had pre-meeting conferences. We've given pre-meeting materials as necessary to him as well. We've made sure at every single meeting that we have introductions at every meeting. And this is, as you can imagine, it's a very large group of investigators here. A lot of this is just done by phone as well too. So it can be kind of hard to figure out who's who. So on top of that, we've provided a bio sheet as well too. So you can always refer to it and see who's actually talking at that point and you know, what they're bringing to the table. And then as well, we've been very good, and Justin's very good about keeping Stu engaged during our meetings as well. So at every sort of point as we start to bring it to a close, if we haven't heard from Stu, we're always trying to ask him, okay, what are your thoughts on this? Do you have anything else to add? And he's been very vocal on some of the aspects, especially of what we're talking about or thinking about for knowledge translation studies. So in terms of what we're thinking about here, little is published in terms of barriers and facilitators for these early phase trials. And we know, again, we've already talked about this, that enrollment depends on the knowledge, values, preferences, and motivations of patients. And we believe that understanding these sort of knowledge, values, preferences, etc., will improve processes to ensure that outcomes address our stakeholder concerns, our patient partner concerns. Also the physicians who are recruiting, their concerns as well. So again, mixed methods, interviews and surveys of patients and doctors. This is grounded in the theoretical domains framework. Just really briefly, what we're doing is interviews. On the top there is interviews of patients to understand their perspective. So a small group of patients, in-depth interviews to understand what the enablers and barriers might be. And then that's followed by a national survey, with a larger group of patients. And we're hoping to distribute that through the Leukemia and Lymphoma Society of Canada. And then on the bottom there is basically trying to understand the barriers and enablers for hematologists to provide ongoing care for these patients and also to recruit these patients into these trials. So again, a smaller number of in-depth interviews followed by a larger national survey of hematologists. And again, as Justin has already pointed out, we're going to triangulate these findings to hopefully inform our clinical trial protocol. So in terms of what this sort of leads to, I'm just going to skip this slide actually because I want to show you practically what this might actually start to look like. And this is sort of some early stuff that we've been looking at for a stem cell trial for stroke. So same sort of objectives here. We're interviewing physicians and patients to identify the barriers and facilitators to conducting a clinical trial for chronic stroke in this case. So some example responses. So we asked, and this is just pilot work here, but what would you like to achieve by participating in a trial of stem cell therapy for stroke? We start thinking about the theoretical domains framework, or what we think this might speak to is the goals. What patients have told us is, I would like to have some form of improvement, even small. Another patient said, I would like changes in my mobility with my spasticity. That's a big one for me for sure. But what this actually sort of points to is what we call the therapeutic misconception. So for most of these early phase and first in human trials, the researcher themselves are not going after efficacy or how effective a therapy is. They're usually just going after whether a therapy may be safe or not. So these very small trials, they can never really speak conclusively to how effective a therapy may be. They can form sort of a pilot experiment almost to make sure that what you're going to be giving, this highly experimental therapy, is not harmful to patients. So obviously there's a bit of misconception here as to what patients are hoping to get out of this to what a researcher is actually gearing their trial towards. So this is something, obviously, this is not new at all. This is obviously something that's been pointed out in other trials and in other domains as well too. But it's certainly something that researchers need to be cognizant of when they start talking to patients about a trial and recruitment for a trial. We're talking to physicians. We asked them, how do you see screening for this trial in relation to your role as a clinician? So they said, one physician, said, it's part of my role in terms of advancing research. And this is not a researcher per se. This is actually just a clinician on the ground who treats patients with stroke all the time. But then we asked them, how often might something else be higher priority than screening for a clinical trial? Well, one physician said 100% of time, and then another one said almost always. So this might speak to some of the issues around why 80% of trials don't recruit, right? So, but this obviously also provides some fodder and some justification when we start to build our trial protocol to say we need to fund research assistants and research nurses on the ground to be doing this, because clearly the clinicians don't have time, right? So, or at least they don't have time within everything else they have to do or their academic and clinical responsibilities, right? So basically all of this work will hopefully, again, inform how we justify the trial and how we're going to run the trial.
I think there's another argument for that intermediary between the doctor and the patient in recruiting, and that's ethics. Sometimes patients feel like they have to go into the trial if the doctor asks them,
right? And
they may not really want to,
right?
But they don't want to, upset their doctor, so they go into the trial and then they don't, stick it out,
right? Yeah,
and that doesn't really help your trial very much.
It's really interesting because the perspective of RABs is that someone within the circle of care has to provide that initial introduction, right? So
I just think that what I've seen a lot of is the doctor mentions the trial, and if the patient expresses any interest in it, then they hand them over to the research nurse or somebody else in the circle of care who doesn't have that sort of quite the same power dynamic. Right.
No, absolutely. Yeah. No. So I totally agree with that. So in terms of take-home messages, last slide here, through GoCart, we believe that a comprehensive research platform that includes patient partners is needed to accelerate this bench-to-bedside translation. I point out just a few of the hiccups that occur that make, that 17 years that Zara pointed out in our first presentation here, and why that actually occurs. We believe that integrated knowledge translation helps patient partners to be collaborators in the research process, and even within the early stages of GoCart, we're definitely seeing that. And then finally, we believe that identifying patient concerns ahead of time may improve our clinical trial protocol and how these trials are actually run.